The human immune system is a formidable protector, but if its targeting mechanics misfire, the network designed to keep us alive becomes a deadly threat. Take the nasal mucosa. Here, mast cells wait on standby. These are the heavily armed sentinels of our innate immunity, packed with inflammatory chemicals and ready to fire at the first sign of danger. If you've been exposed to an allergen before, your body built a specific warning system. It manufactured IgE antibodies, specialized proteins that permanently attach to the outside of the mast cell, coating its surface like tripwires. Now, a harmless environmental intruder, say a grain of pollen, drifts into the mucosal space and bumps into the mast cell. To avoid misfires, the trigger requires a precise physical connection. The pollen grain acts as a bridge, locking into two adjacent IgE antibodies simultaneously. This physical cross-linking throws a biological switch, sending a signal deep into the cell to dump its payload. The mast cell degranulates, violently expelling histamine and other mediators into the surrounding tissue. These chemicals force local blood vessels to widen and leak, rapidly generating swelling and inflammation to flush out the intruder. This localized hypersensitivity is a false alarm that causes a stuffy nose or hives. But when these same targeting errors move from the surface into the deep bloodstream, the stakes shift from temporary discomfort to widespread organ damage. Let's move away from the mucosal surface and drop into the high-pressure environment of a medium-sized artery. In a condition called polyarteritis nodosa, the immune system launches a catastrophic assault on the body's own infrastructure, and it happens entirely because of passive biological debris floating in the blood. When the body fights a virus like hepatitis B, it produces antibodies to neutralize the viral antigens. These antigens and antibodies bind together to form immune complexes, heavy, tangled clusters of molecules that circulate through the vessels. Eventually, these complexes grow too large. They sink out of the blood circulation and embed themselves directly into the delicate tissue of the vascular wall. This trapped debris trips an alarm. The complement system, a cascade of plasma proteins, activates. A specific protein called C5a acts as a chemical beacon, summoning heavy immune artillery to the site. Circulating white blood cells called neutrophils arrive to clear the mess. They attempt to swallow the immune complexes, but because the debris is firmly wedged into the vessel wall, the cells can't ingest it. Biologists call this frustrated phagocytosis. Unable to engulf the target, the neutrophil resorts to a destructive contingency plan. It forcefully expels reactive oxygen species and highly corrosive lysosomal enzymes directly onto the vessel wall. The collateral damage is total. This chemical bath completely dissolves the structural integrity of the artery, causing a pan-mural destruction known as fibrinoid necrosis. The immune system destroys a perfectly healthy, essential blood vessel simply to eliminate a trapped piece of biological garbage. We trace the vascular system even further down, arriving at the microscopic capillary beds of the kidneys, known as the glomerulus. Diseases like granulomatosis with polyangiitis present a paradox. Even as the capillary walls are being destroyed, an examination reveals they are completely clean, with no trapped immune complexes anywhere to be found. Instead, the error begins with a molecular anomaly. Environmental triggers cause neutrophils to push their internal enzymes, specifically PR3 or MPO, onto their exterior surface. These exposed enzymes attract a rogue element. Autoantibodies known as ANCAs emerge and lock directly onto the misplaced enzymes. This immunofluorescence image shows the diagnostic fingerprints left behind. Medical investigators use this stark, glowing pattern to identify ANCAs binding to neutrophils. The moment ANCA binds, it bypasses internal checkpoints, forcing the cell into an active attack state despite the absence of any trapped complex. The hijacked neutrophil leaves the bloodstream and burrows into the completely clean, healthy endothelial wall, tearing the tissue apart from the inside. After this unprovoked attack, the degranulated white blood cells dissolve into the ruined vessel, leaving behind a microscopic trail of debris that pathologists call nuclear dust, or leukocytoclasis. To stop vasculitis, medicine must identify the exact pathological fingerprint driving the damage. Whether it's IgE tripwires, trapped immune complexes, or hijacked ANCAs, Understanding the precise mechanical error is the only way to halt the friendly fire.